Okay, I think the numbers are starting to stabilise now. So I'd like to thank everyone for joining us for the latest presentation in the Advanced Plant Growth Centre seminar series. And it's a great pleasure to introduce today Professor Gail Taylor, who's currently at the University of California, Davis, where she's a distinguished professor and John B. Orr Professor of Environmental Plant Sciences. Gail's moved to California in 2017, and prior to that, which is where we met in a previous life, Gail was a professor at the University of Southampton. She's done an extensive amount of research into leafy greens, the genetics of leafy greens, and also energy crops and particularly energy trees, which we'll hear about now. So I'll stop and hand over to you, Gail.
Thanks, Rob. That's fantastic, and welcome everyone, and it's a pleasure to be here. Today I'd like to talk about one half of my programme, which is clean, safe and sustainable leafy greens, moving towards indoor systems. And I'd like to acknowledge California Leafy Greens for funding, and also the AI Institute for Food Systems, which is a new NSF $20 million institute, which we're developing on the campus. I've got lots of data, so I don't want to get too out of time. So I'd just like to focus today on the work we're doing on nutrient-dense watercress breeding and also the lettuce microbiome. We've got some interesting stuff going on there. Very happy to have a conversation with people afterwards, either in this session or please contact me at UC Davis. So I always like to start by acknowledging the amazing people who have helped me over the years. And here we've got 10 incredible women: Libby, Hazel, Annabelle, Yufei, Gaia, Nicole, another Libby, Susie, Adrienne, and Lauren. And I suddenly looked at this last night and thought, oh, we've got some gender balance issues here. But it wasn't deliberate that we appointed 10 women that have been working on these projects over the years. But I probably should write a story on this. I think it's fairly phenomenal. But not to forget that the person who kicked off the leafy green research in my lab was Dr. Graham Clarkson, who's now the strawberry breeder for Ed Vinson. My work over the years has been funded by BBSRC, now the leafy greens in California, and also Sainsbury's, and not forgetting 20 years of funding from Watercress in the UK. So thanks to all these amazing people for doing fantastic work. So why leafy greens? Why am I so interested in leafy greens? Well, in California alone, the lettuce market's valued at around $3 billion per year. We grow 85 to 95% of the lettuce for the whole of the US. And the growing moves from Salinas Valley in California to a valley in northern Arizona. So summer California, winter Arizona. Lettuce is widely consumed across the world and consumption is increasing. So if we look at some USDA statistics, the average US citizen these days eats about 12 pounds of lettuce a year. So it's everywhere. It's on the side of a plate, it's in main meals, it's in Western cuisine and Asian culture. And this lettuce market in California, it's about the same size as the wheat market in the UK. So it's significant, it's not going away. And so even in indoor growing systems, we shouldn't forget that it's an important set of foodstuffs. What about the challenges and opportunities then? So challenges here in black, and some of the opportunities that maybe morphing to an indoor system might offer us. So number one here, crop nutrition and quality. In breeding, these have traditionally not been a focus. Particularly in leafy greens, it's been about yield and disease resistance, as in many other systems. Is this important? Can we improve it? I'm going to talk about that today. Leafy greens consume significant amounts of water. I'm not going to talk about this today, but we know these indoor systems offer huge advantages. So 95% reduction in water use when we grow crops indoors, such as here. Foodborne illnesses are a big problem with the consumption of raw leafy greens from outside culture. And we're interested in particularly harnessing the microbiome to address this. I'll talk a little bit about that. And then more generally, I'm going to talk a little bit about breeding and what this can deliver. So this question of nutrition, we talk about food security often, and it's largely about calories, so the calorific value of food, but I'm very interested in nutritional value of food. And FAO published this report a couple of years ago in 2019 about the future 50 foods that could feed the world to make healthier people and a healthier planet. So these future foods were chosen based on their high nutritional value, low environmental impact, flavor, accessibility, acceptability, and affordability. And here's the list that we have. And I'm delighted to say there are a number of things here that you might imagine, such as beans and pulses, mushrooms, but leafy greens are one of the top categories to feed the world in the future, because potentially we can pack them full of nutrients and make them nutrient dense. Is that what we've been doing with our foods over the last, let's say, 100 years? Well, it turns out we haven't been doing that, and we can find lots of data. These are actually published in the New York Times. And they show the progression of breeding from, in this case, the wild crab apple to the modern day apple. And I was literally last week looking at similar data sets for strawberry, where in California over 100 years we've increased yield and decreased the phytonutrient profiles that are linked to flavor. So it's well recognized that here total phytonutrients have declined as we've gone into modern varieties. And similarly, if we look at the leafy greens, this is amount of antioxidants per 100 grams of fresh weight, and we can see it's declined as we've gone from sort of wild relatives like dandelion through to iceberg lettuce, which is hardly worth the jaw action to eat. It's such a poor nutritional crop. And I'm delighted to show this. This is some of our own research, which we did in collaboration with the Hutton, with Rob Hancock some years ago. And this is a phenolic profile for our wild lettuce, Lactuca cereola, and Lactuca sativa, the cultivated lettuce, which we were crossing to get our biparental mapping population at that time. And you can see a really good profile of how we have we've simplified the complexity of the chemistry of these leaves has been reduced as we've bred. And the picture's complex, but it does suggest that we might be able to harness some of these nutritional aspects of lettuce for the future. In our other research, we're trying to use predictive breeding to do this. I'm not going to talk about that today. It's something that's ongoing now, but Dan Runcie, my colleague, is very interested in how we can define good nutrition, what it means, and what we should be breeding for. So this was some work we did with Rob. And the problem is, if we're interested in the genetic basis of nutritional quality, this was 15 years we took to get to the target. And in that research of Annabelle Dameron with Rob, we managed to identify the QTL using this mapping resource. We identified the target. We made a molecular marker and we used it to select some better lines, and we have crossed those back into some commercial material. So we now have high antioxidant status lettuce. So we made a significant improvement, but it took a long time. So we're all familiar with this on this call, that breeding takes a long time. So here's Richard Mitchellmore. He developed the original cross, I think in the 90s or even earlier, between the wild and the cultivated. He got it down to F9. So I go to his lab now. He's in Davis at the Genome Center. Can get some of his F9 seed. We've grown them all over Europe and in California multiple times to get to that target and do molecular breeding. Very hard work. So plant breeding is traditionally slow, and this is the central dogma that we're all familiar with. So we make crosses, we get genetic variation, we select, we get new varieties. And so the thing about indoor ag and leafy greens, which is one thing I'm particularly interested in at the moment, is can we speed up that process? And we do fast-track bespoke breeding. And yes, this should be possible. Why? Because we know that traits are determined by genotype, the effect that gene exerts over a trait, but also the environment and the management, the agronomy traditionally. And this is like the bane of the plant breeders' life, in that these G times E interactions, when you get to your final sort of proof of concept and you do multi-site trials in multiple environments and you're trying to harness the trait and introgress it, this can be really tough and slow. And of course, now we have these perfect environments. This is my container farm on the UC Davis campus. It's a 40-foot commercial container, which is pretty good. And you can see us here growing one of our favorite crops, watercress. So we can now do, hopefully, bespoke, targeted, controllable breeding, where we can match the traits of interest, nutritional traits perhaps, to the particular environment and make really fast progress. And if we harness this to biotechnology through gene editing, and also that predictive breeding through AI using genomic selection, the idea is that we can make really fast progress. And this is really where my research is at the moment. So we've made some limited progress to date, and I'll tell you what we've been doing. But indoor ag then, is it a high-tech or a low-tech solution? And we often wrestling with these concepts, and I'd like to say it's both, and we can think of it in these two ways. So it might be the high-tech solution for personal health, premium organic food delivered to Western urban populations, but it also has a significant amount of power in delivering nutritional value, focusing on the circular, renewable economy. It's sustainable, it can provide local employment and community food. And we're interested in both of these aspects of the research. So for example, in that Western high-tech solution, we know that diet here is important, and we can link it through to nutrigenetics, nutrigenomics. We can imagine a world in the future where we're all sequenced and where our dietary requirements are linked to the needs we have for our microbiota, for example, and then particular epigenomics that we might want to deliver this personalized health. And this isn't too far away from reality, and we can debate whether it's blown up out of all proportion or whether in future this will become routine. At the other end of the scale, of course, this is one of my colleagues, Alan Van Duyn. We're breeding orphan African crops, and many of these are leafy greens, for healthy diets, nutritious local crops to deliver the food of the future on the African subcontinent. And I would argue that this indoor ag has a role in both of these, and we're looking at ways to make it happen. And we're at the beginning of that journey, make no bones about it. So why is this my favorite crop? So this is watercress, Nasturtium officinale. It's a great crop. Why is it my favorite crop? Well, it's the most nutrient dense leafy green on the planet. It's the powerhouse fruit and vegetable. So in an assessment of 17 nutrients, and this was published by Denoa in some years ago now, 2014, watercress beets Chinese cabbage, charred beet greens, and all these. And you can see that the dark green leafy greens go down to the lighter greens and through the reds and the lighter greens, but watercress beats them all, and it significantly beats kale. So it's a way we can pack in nutrients and make a nutrient dense crop. I'll also argue it was the first fast food. So in the US, I like to tell people this, that, you know, fast food was invented in the UK. Watercress was the food of the poor agricultural workers, grown in diverted streams. You can see it here, where wells were dug to get the water that was needed for this aquatic crop. And then when the steam trains came to rural areas, women usually took baskets of this food and took them to London and served the bunch of watercress with a hunk of bread as the first fast food. But so it has a long cultural heritage, but virtually no breeding and resources, no selection and crossing, no molecular biology, and no genome sequence. So over the last 10 years or so, we have been addressing these limitations. And although not all of this work is published, we have actually been able to address all of these limitations. We're in a really good position now to move forward with watercress, but we have been developing a lot of these genomic resources. So watercress, then, it was the first fast food. It's a crop of the past, but does it have a future? Well, yes, it's a fabulous crop for indoor ag because it's generally an aquatic, a semi-aquatic plant. It does really well in hydroponics. So, for example, this is Aerofarms here. It's one of the largest east coast of the US indoor farms. And the CEO and founder, Mark Oshima, was in the department last week, and he kindly flew some watercress all the way from Newark on the East Coast so we could eat it in California. It's a very good crop. It comes in a baby leaf and one of their shoot mixes, and it is being grown commercially. But it's largely unimproved and not bred for these indoor systems, so that's where we come in. So it's a brassica, and we know it's proven that these consumption of these crops, all of these crops, is linked to the risk of cancer, reduced risk of cancer, cardiovascular disease, diabetes, and a whole range of other diseases. And probably you're familiar mostly with broccoli and the Beneforte broccoli that's been released by JIC and the institutes down in Norwich. So we know that consumption of these crops is really good for human health. Watercress is Nasturtium officinale. It's a perennial dicot, part of the Brassicaceae and semi-aquatic, and grown in these traditional beds in southern England. It has a very long history and lots of published research to support these claims. And one of the important things, again, many of you know this, is that the glucosinolates in watercress, its gluconasturtium, are hydrolyzed on the production of myronase following chewing, which mimics insects. And we have this chemopreventative phenethyl isothiocyanate, which produces the peppery taste in watercress. As I say, many of these pathways are well known, and in my own research, I've been working on breast cancer cell lines with Graham Packham, where Graham has shown that the tumor angiogenesis, which is linked to cancer metastatic tumors forming in these breast cell lines, is blocked by phenethyl isothiocyanate in vitro studies. This HIF factor blocks the angiogenesis pathway in these cell lines, which is great. So we have some understanding of the mechanism of action. So what have we been doing then on the plants? Well, we took benefit of a collection that Steve Rothwell from Watercress have been seminal in collecting from all over the world. So we harnessed as much natural wild germplasm as we possibly could, and we have up to 50 wild accessions of germplasm. We've done a lot of work on these over the years, and here's one figure for antioxidant capacity, and just looking at 18 of those accessions to show that they vary significantly. So there's a lot of variation there, and what we did was we selected 2 of those wild accessions, and we've been involved in making a cross. We made an F1, and we made an F2, and we did DD Rad with Ed Buckler before he got closed down, and made the first watercress mapping population and rills. And this is where we're at with this now. So we have this biparental mapping population with an F1, and we've been doing single seed descent through this orange colored line here. So we're actually down to the F5 now. So we've significantly reduced the heterozygosity, and we're about to resequence this to do a sort of genomic prediction study so that we can take some of our lines that I'll tell you about in a moment forward in a breeding program. And we've also done some what we call mini bulk populations where we can start studying some of our traits of interest. So they have a reduced heterozygosity, but they're not as good as the single seeds. But we use them in our current studies. So this is the research of Yufei Chan and also Lauren Hibbert. So what are the things that we're interested in? Well, we're interested in these secondary plant metabolites, so the antioxidants and glucosinolates. Believe it or not, we're trying to improve the flat sweetness of watercress, because the perception of watercress is it may be a bit too peppery, particularly for the less mature people of the world, shall we say, that their palate is rather different. And so We spend a lot of time actually tasting watercress. So some of my colleagues on the UC Davis campus, their research is about tasting wine, making wine, going to the chateaus of the world and tasting wine and training winemakers of the future. But my lot is the watercress, which I'm delighted with. So we're interested in flavour and chemistry. We're interested in this forkability, so plant architecture, essentially. We also have a project on resource use efficiency, where we're improving nutrient use efficiency, but I won't talk about that today. So all of these resources that we've been making are going forwards to these targets. So as with many things, we've gone through 3 versions of the map. So we had an RQTL map. We got to join map, where we resolved it to 16 linkage groups. And the best map we have, which is still in progress, we've harnessed the watercress genome, and the genome is looking really powerful now. But at the moment, we still map QTL with this version 2. So we've done a lot of the work, the only watercress map in the world that we've developed these resources. And moving on to the genome, when I moved to Davis, the Genome Center helped me to sequence the genome. So we did some 10x sequencing, and we produced a really nice map. Now, I'm not an expert in this area. I was helped by a number of colleagues here, but I'm reliably informed that these assembly stats are okay. One of my colleagues then stepped in, Ming-Chao Liu and Ting-Ting Zhu on the campus, and they thought, following a seminar, that they could significantly improve our genome. So they improved the assembly using this bio-nano technology, which we have here in the department. And again, some very good statistics. They were able to resolve a lot of misassemblies that we had in our 10x PacBio data. And we have this beautiful resolved genome now, which we're just working on publishing, which has 16 linkage groups, which is great, anchored on 59 scaffolds, and a really good genome to be working with. So what have we done with that? Well, when we were still based in the UK, we have grown this genome in the outdoor traditional watercress farm. And we've also grown it in indoor controlled environment facilities. I'm sorry I don't have a picture of that. And we're currently growing it in the vertical indoor farm. So we started to understand what we can lever from this population. We've done lots of phenotypic mapping. Here's some examples. We've got parent A and parent B here, which, if you remember, we took from the wild accessions in blue and red. And you can immediately see we have lots of transgressive segregation, which is absolutely wonderful, and lots of phenotypic variation, typified here by a few images of what we see when we grow this material. As I say, we've worked on this material in both indoors and outdoors. Do we see some differences? Yes, we do. But we have this transgressive segregation, so wonderful things we might be able to harness for the future. So, for example, specific leaf area, the sort of thickness of the leaves is a nice one that we can harness for the future. But I just wanted to point out one set of data in a little more detail, in that we did with Graham Packham this cell cytotoxicity assay using human breast cells. This is a first for us, and there are not many studies like this, and these are the cells here on the right. This is the research of Nicole Vucina, who now works at the University of Exeter, and she looked at the metabolic activity, so if you like, the killing capacity of our extract of watercress juice, and this is the concentration of watercress juice, and this is the metabolic activity. So we often go to 50%. How do we kill 50% of these breast cell lines? And we look at the concentration required. What you can see here in the black, red, and blue is that the black is the real population, some selected individuals, and the red and blue are those parents that we selected. So you can immediately see that we have differences in the red and blue lines. That's really interesting because we've unleashed some genetic variation, which maybe we didn't have previously. And if we look at the whole population, which is here, so this is the IC50, the killing capacity of watercress juice. Now, I didn't tell you before, but one of our parents that we selected is very close to a commercial parent that, sorry, the commercial line, which is extensively used in the UK. And this is where the commercial line sits for IC50. So anything left of this line means that we've improved the anti-cancer, the ability of this watercress extract to kill these cancer cells. Now, we did over 1,000 watercress samples. So we did this in triplicate on our mapping population. We have transgressive segregation, and we've mapped this trait. And here's Nicole doing this analysis. Okay, it's in vitro, it's not the whole story, but nevertheless, it provides a useful target for breeding. What were we able to show? Well, when I moved to Davis, again, Dan Klebenstein is a sort of pretty much world expert in glucosinolates, and he has a really good setup, and he just took my population and since then has just done thousands of assays for us. But here's one result, and this is one of the linkage groups of watercress. And we can see here at the far right, this is the IC50 QTL, which is mapping here. And this IC50 is mapping alongside several for long-chain glucosinolates and also in watercress for the phenethyl isothiocyanate. So we have a really good target here in the watercress genome for breeding enhanced anti-cancer activity in watercress. It's not all of the story we know, but it's a great target. What about indoors then in the true vertical indoor farm? Well, I wanted to just tell you a little bit about some of the work, comparative work we're doing on watercress. So one of the first things we did is we took the commercial line, which is the Watercress line, which they've been inbreeding for about 35 years, and we grew it in a traditional UK stream here. This is the bed. We also grew it in California in traditional irrigation ag in California. And then we grew it in the vertical indoor farm with less and more amounts of blue light. And we looked at yield, crop morphology, and these anti-cancer glucosinolates. And we published this recently. And in bringing these studies together, we wanted to know what was happening to yield, for example. And so you can see here the California crop, the UK crop, and the two vertical indoor crops, low blue light and high blue light. And we look at various traits. One we're particularly interested in, though, for growth is this one. It's leaf area. Leaf size is a key trait. And you can see in California, although we had a lot of yield in stems and dry weights, for leaf area, this wasn't so good. So to put it bluntly, in California, we produced a very stemmy, sort of fibrous crop, which we weren't sure would have much commercial value. It may be of value in the Asian cultural market because their watercress is used in soups and stir fry. Higher yields in the UK, but what we quickly discovered was that in our indoor vertical ag system, we could beat the yield. So we were able to grow a really good large leaf crop in these indoor systems, which is fantastic. But most exciting of all is that we looked at these glucosinolate compounds with Dan in California. Lots of glucosinolates, perhaps reflecting the hot, dry climate, quite stressful climate that these plants are subjected to. Some glucosinolates in the Ukraine crop, but in the indoor vertical crop of grain, we were surprised to see enhanced glucosinolates. But not only that, when we look in more detail at the profile, we've actually found a new glucosinolate which doesn't appear in our system at all in the UK or California. So we've altered the quantity and quality of the anti-cancer chemicals. So we believe this means there's lots of potential to breed in these systems in the future. Where are we at the moment? Well, this is about 30 of our wild accessions, which we've currently exposed to our control treatment and our additional blue light treatment in the indoor farm. And this red line here is the current commercial line, which is grown in the UK. This BMW is a Florida line, actually. So that's the red line, the yield, which is here. we call it Vita, WX Vita. So two things are apparent from this figure. First of all, we have massive variation in the wild accessions. This is fantastic. So we're planning to start making crosses from these high-yielding genotypes. We have the samples for the glucosinolates, but we haven't looked at them yet. And the second thing to note is that blue light has interactive effects on this material. Sometimes it stimulates yield, sometimes it reduces. And this is, again, dry weight. We're still analysing our leaf area data. So lots of variety, lots of potential to breed, bespoke breeding for the indoor environment. So that's where we're at with watercress. And so we're moving forward with our indoor breeding program, looking for these bespoke traits. I mentioned safety, and I wanted to talk a little bit just before I wind up on a second project, which is about making lettuce clean and safe. So we could title this The Microbial Life on the Lettuce Leaf, and how we can look at the extended phenotype between leaf surface traits and the abundance and diversity of the leaf microbes, the microbiome. And over here on the left, this is the Maricopa Arizona rig. It's the Lemnitech high throughput phenotyping rig. And of course, this is how, so we do some work. We have a multi-state project with University of Arizona, Duke Pauly, he's a great guy, and Richard Mitchellmore. So this is traditionally how we grow a lot of lettuce in these big lettuce fields, and you can see here we're doing some phenotyping. So a lot of harvesting, a lot of humans in the field, human interaction in washing and packing. So we can think of these leaves they're in a complicated outdoor system and a cold chain, and they are eaten raw. And this is how many of us consume our baby leaves at the moment. And so this presents challenges for food safety and also for minimizing spoilage. We've all had that experience where you take your salad pack home and you end up with a slushy mess in the bottom of the fridge. And this is the target of our research that's been ongoing for a decade. We're doing lots of things on spoilage and shelf life. And I wanted to just focus on one aspect of that. And the aim of this particular project is to investigate the potential of the bacterial microbiome on leaves, that we can sort of harness the biology and genetics rather than washing and processing. So all of this is designed to wash off those microbes that get onto your leaves when we grow these things in the field. And these microbes can be very damaging. So we know that lettuce consumption, particularly romaine here, is linked to recent foodborne E. coli outbreaks. E. coli, Shigella, Salmonella, Clostridium, Campylobacter, and Listeria. These are human pathogens that get stuck onto these leaves, and they're really, really hard to break off. And I don't have a slide of it today, but they get into this biofilm, this ecosystem on the leaf. They stick to their leaf, they go into a viable, non-culturable state, and they spring back to life when you get them in your fridge. And so it's really hard, whatever washing is done, to really remove them from the leaves. And this is a very active area of research. And of course, I just wanted to remind us all that down here, this is how the world wastes food. So this is how many calories we waste every day, per person per day. So in the US, it's a staggering over 1,000 calories a day we waste. This is largely with the consumer. And even in Europe, it's 750 calories a day. So this is through spoilage. And then as we go to the developed world, it's largely through breakage in cold chain. So there are great reasons for wanting to understand what's on the leaf in terms of the microbiome. So we have all sorts of microbes that sit on your lettuce leaf. Don't want to upset anyone here, but there's a lot of them. And as I say, they're very hard to release from the surface of the leaves. They can be pathogenic to plants and people. They cause spoilage, but they can protect plants from disease. They can increase yield or decrease yield. So we want to understand them. We want to understand how they control plant disease, how they interact to cause these human pathogen outbreaks and spoilage. And as many of you may know, these microbes slug around in the environment and they get onto the surface of the leaves, and this depends on the climatic factors. It depends on colonization routes, the types and number of microorganisms present, and then it depends on the surface of the leaf. And these are some of the things we're interested in. It depends on the epidermal cells, what they look like, how many junctions there are, how the stomata formed, how big they are, how frequent. This is a major pathway. It depends on signaling molecules within the leaf, and we're interested in all of these things. So how can the leaf microbiome be manipulated? Well, we could be interested in the colonization routes, the climatic factors, but as I say, we have been focusing on host physiological traits that affect attachment and proliferation. And this is largely the work of Libby Arnold, a PhD student, and Annabelle Dameron, who came to run my lab in California. What are the questions? Well, can we breed for a good microbiome? Can we manipulate these leaf microbes to help prevent food spoilage and human pathogen outbreaks? How does plant genotype affect microbial composition? How do these morphological traits affect microbes? Can we quantify the genetic architecture of host and microbial traits and identify candidate genes in the host lettuce microbiome that we can then breed for? So this goes back to Dawkins' concept of the extended phenotype. So it's nothing new, but deploying it in breeding is pretty hard because quite a lot of the seed houses aren't really very interested in my leaf surface quality traits. But these are some of the things we're interested in: stomatal density and index, size, length, width and length, how the epidermis comes together. Here you can see, I've taken from the literature, a big open stomata, lots of microbes getting in, and these cell edges where the microbes preferentially proliferate. We're also interested in leaf hydrophobicity, waxes, and we measure that using contact angle. We drop a controlled droplet of water onto the surface of the leaf and see what angle it makes with the lettuce leaf. So over the years, we've been looking at UK grown salads. We've looked at different species in the UK, lettuce, spinach and arugula, rocket, and looked at how they vary. So if you go to an average salad field, do a big harvest, what microbes do we see? We've done a study in Portugal. This is using our biparental mapping population. Since moving to California, we've moved to a GWAS population where we're examining G times E interactions and mapping these traits. But today I just wanted to tell you a little bit very quickly about some of our biparental mapping population data. And we have these in bioRxiv now, and they're just in, we're just doing some reviewer comments for publication. So we've scraped the DNA off the surface of the leaves and sequenced it and looked at, we actually do metabarcoding. So we look at the hypervarial 16S rRNA. We do this, we know how to do it. Yeah, it took 6 months to figure it out, a good technique, but we can harvest the leaves, get the DNA, do, as I say, metabarcoding, and identify the microbial bacterial species on the leaves in all of the genotypes of a mapping population. If you look at our two parents and we compare our wild lettuce and our cultivated lettuce, so these are the two parents I've mentioned before, we can see that with breeding, we've developed larger leaves, good for yield, smaller cells, fewer larger stomata, that could be bad for microbes, a lower contact angle, likely that the leaves are more wettable and less hydrophobic, and we've got these traits here. So we've altered surface properties, and in our biparental mapping population they differ. These are different ways then of looking at the microbial data on the surface of the leaf. So one that you may be familiar with, the Shannon Index, looks at beta diversity. We can see that in our cultivated population, we've probably reduced the diversity of the microbes, the bacterial microbes on the surface of these lettuce leaves. And then if we do some KEGG analysis on this data set, we can see the things that are in higher abundance on the cultivated leaves. Interestingly, some of the plant pathogenic signaling molecules we see and quorum sensing are there, and some aspects of the metabolic pathway. So these are all the things that we might delve into more deeply for our breeding targets. But what we also did is we took the whole population of 171 individuals, we sequenced the DNA on the surface of the leaf, and we looked at relative abundance, and we looked at these ASVs, so these sequences that are really distinct sequences that we can map to the databases for our microbes of interest. We looked at alpha diversity, 4 beta diversity traits, similar ways of looking at beta diversity to the Shannon Index, and we looked at what was present. And you don't have to understand this figure here, it's just to say that we've tried to map our leaf surface traits to our microbial traits and looked at PCA for these genera of interest across the mapping population. And you can see on this top part of the figure, this is the microbial relative abundance. What you don't have to read any of the details here, you can just see it varies across the population for these 171 individuals. So what we've been able to pull out is significant associations between the bacterial community structure and key traits of interest on the leaf surface. Perhaps not surprisingly, those are stomatal size, leaf perimeter, and epidermal cell size and number, and we can see this in the PCA. And here we have some of the statistics. Do we have human pathogens on our population, which was grown on a commercial farm in Portugal? Yes, we do. We see Shigella, Clostridium, Campylobacter, and Staphylococcus in smallish numbers, but they are nevertheless there. So finally then, I wanted to finish on this figure, which is a little bit complicated, but we've mashed all these data together. So we have, for example, in green here, this big block in green, this is the lettuce genome. So we have linkage group 1, 2, 3, etc. through to 9. So we've got the lettuce genome here, and these green traits called on the left and shown here in green are our surface leaf characteristics mapped as QTL from our biparental mapping population. So you can see on linkage group 1 we have a few traits. We have a really big hotspot here for some of these leaf surface traits on linkage group 6. So next then we've mapped alpha diversity, which is in the middle here. Well, that's just the number of genera that tend to be in the bacterial microbiome on the surface of the leaf. And then more importantly, we've mapped beta diversity, which is this pale blue circle here. We have some traits mapping. And perhaps most importantly, we've mapped the individual bacterial components of the microbiome to see where they line up on the genome. Now, for us, we've identified several areas of the lettuce genome which will be informative in breeding, which will enable us to manipulate the microbiome that might sit on the surface of these leaves, and that we could manipulate to improve the ability of these leaves to dispel human pathogens. So a couple of these, I'll go over 3 in a bit more detail. So linkage group 1, this happens to be an area of the lettuce genome where we have stomatal length and width mapping. So we have a QTL here, but I haven't gone into this data in detail, but we did a metabolic KEGG analysis. And this here is an area that maps to some core genera that are hubs in our hub and spoke network analysis that we've done. So it's an area of the genome of interest, and we're not quite sure why, but we want to look at it further. This is an interesting area of the lettuce genome on linkage group 5, because it happens to map to a stomatal trait. but more importantly, it maps to this, this Pseudomonas genera here. Pseudomonas are really important in lettuce for spoilage, and we've got a lot of information on various Pseudomonas, but here we've got one mapping to this QTL that maps to a stomatal trait of interest. It also maps to this human pathogen. So this is E. coli that's mapping to this stomatal width trait on the lettuce genome. Now, most interestingly, perhaps, is this area of the genome of lettuce on linkage group 6, where we have lots of co-locating, perhaps, link traits. So we have stomatal width, stomatal length, stomatal index, so the number and proliferation of the stomata. We also have contact angle, that hydrophobicity, epidermal cell perimeter, the number of edges, so the edges relative to the volume of the cells. So this looks like a really interesting area of the leaf surface traits, the genome of the lettuce. And not only that, it maps to several of the microbes that we see on the surface of the leaf. And this includes 2 which are human pathogens. So that's Clostridium, and also there's another one. I can't remember which one it is, but we have another one mapped to this area of the genome. So our future research is targeting these areas of the genome. As I say, at the moment we have 2 GWAS populations which are being grown in Central California and Southern California, where we're doing a GWAS analysis of the lettuce leaves and microbiome analysis to try to unravel these traits a little bit further. So if I could conclude then, I hope I've shown today with a little deep dive into some aspects of my program that indoor ag systems offer potential for fast breeding and the capture of these novel traits for particular bespoke environments. And we're starting to see lots of examples of that, with some focus at the moment on blue light. We've established a world-leading, I'd like to think, watercress breeding program. We're moving to indoor ag. It looks promising for manipulating anti-cancer chemistry and other traits where, as I say, blue light seems to be important. The wild accessions of watercress are showing wildly varying response to blue light and also yield. We're looking at them actively. The lettuce leaf microbiome varies with leaf genotype in mapping and GWAS populations, and we're trying to figure out what the breeding targets are. I haven't spoken about it today. It's a little bit tricky because I'm under NDA on this. I'm working with an industrial collaborator, but the microbiome of the indoor vertical farm for leafy greens is staggeringly, excitingly very different to what we see in field-grown leaves. And it gives me the belief that we really are growing squeaky clean leaves in the indoor systems relative to what we've discovered outdoors. And we've managed to correlate leaf traits to the presence of different bacterial genera, including stomatal size, length and width, epidermal cell patterning and cell number. And we've identified these 3 areas of the lettuce genome that look really important for future breeding. And we hope that they'll be able to deliver improved food safety and reduced lettuce spoilage. Thank you very much for your attention. I'd be happy to take any questions.
Thanks for a fascinating and very wide-ranging talk, Gail. So if anyone would like to ask a question, they can either just unmute and dive straight in, or alternatively, if you're feeling a little bit shy, feel free to type it into the chat box and I can ask the question on your behalf. And I see Derek's straight in there, so Derek, go ahead, please.
Thanks, Gail. That was really enjoyable. It struck me that if you're looking to design safer plants, and if you're talking about these adhesion points between cells, if you're going to start manipulating that, presumably it isn't going to taste like what it was previously. The mouthfeel, the organic bit. So everything's a trade-off, I guess.
Huge trade-offs. This is such a good point. You know, any of you in the leafy green business know that red leaves a pack full of anthocyanins, so they're really healthy and they have all those secondary chemicals. However, their shelf life is really poor, so they're always the ones that sort of flop down and look terrible. So very early on in some of our research, we were able to grow really deep red leaves. They had fantastic shelf life, and I thought, oh, this is really easy because we've made these long shelf life leaves. The trouble is, they tasted so bitter that no one could really eat them. And, you know, that was a problem with some of the early broccoli, that it can be made over bitter. So I would say that it's something we have not paid enough attention to, these trade-offs, because we're dealing with a lot of the secondary chemicals that might be signaling and doing all sorts of things. It's been really hard. So our current Watercress experiment, we have a taste panel set up, and the next one we do in our indoor farm, we're also hoping to do a taste panel. I entirely agree, but it's been a bit beyond me because it's so expensive doing some of these panels with humans. But it's a bit easier in California on the Davis campus. So we're moving into the Mondavi Wine Center for their sensory analysis lab. So, you know, as I say, a lot of my colleagues are there tasting wine. We'll be on the watercress and lettuce, but hopefully we'll be able to get to some of these answers. So totally agree. It's all trade-offs and we need to understand and manipulate them.
I think putting it through to high throughput phenotyping, so identifying key organoleptic indicators. I mean, the glucosinolates, you can wallop them through an MS fast, but there's other indicators that I think at least you would narrow down on that basis before you went to an expensive person.
Exactly. So that's what they're trying to do with strawberry at the moment. But these profiles are pretty complex to, from my look at them.
Yeah.
Maybe the human palate is a better thing.
Oh, it is. I mean, the human's the best computer you can get, but I think as the data management piece comes in, and there may be people in the audience that can comment on that, I think it's mining that more sensibly. Whether it's
Totally agree, and that's what we're trying to do in this new AI institute, but I haven't seen it yet. But yeah, it's the future. It's very exciting.
So I believe.
Raoul, had you raised a hand?
Yes, I put my question there, but to keep supercharging the leafy greens with nutrients, so where do you see the real potential about between gene editing versus the breeding approaches that are the approaches that you show us? So do you believe they have limited technological limitations, or we are lacking of basic knowledge to modify the pathways?
Well, I mean, technological limitations. I mean, I didn't speak about it today, but we've made both RNAi lettuce and we've gene edited lettuce. So on that biotechnological front, you know, we've done that. We've done amazing things. We increased the shelf life of lettuce through targeting a cell wall enzyme, xyloglucan endotransglycosylase. We have really strong data on that. So that works. So is it a technological limitation? Not really. Gene editing in lettuce is sort of routine now.
Mm-hmm.
Understanding what to target, you know, there's now the concept that we can pyramid things and do more of that. But a lot of the traits that I'm interested in are complex traits. And so I guess I'm more going towards genomic predictions because they can be so powerful for some of these complex traits, because we can look at, you know, bunches of markers across the whole genome. So we're doing both, but, you know, what's limiting us? Well, of course, social license to operate. There hasn't been, I could give you gene edited lettuce in a few months. But would it be, who would buy it? Who would be marketing it? Who would be, you know, but so the way we tend to see that, it's a great proof of concept. For lettuce, in particular, we have so many targets because it's such a low starting point. Because it's really poor in nutrition. And we've bred all these bad things into it. It's great for yield, but you know, we've done a lot of bad things along the way. So it's a low starting point. We can make big progress. But when is the public going to accept eating raw gene edited leaves?
Thank you.
So, Gail, maybe I could come in with a question there. So, I mean, you know, you've made this observation that in breeding, we've seen this kind of loss of phytonutrients. Now, clearly that hasn't been directly selected for. I guess it's maybe been indirectly selected. And do you have any insight as to what traits might be the traits that are being selected for, that are being co-selected, as it were?
Well, that's a tough question. I mean, basically, we you know, go back to fundamentals. There's this growth defense balance concept that we're all familiar with, that if you grow plants in luxurious conditions, they put everything into primary metabolism and photosynthesis and growing, and then if you put them in stressful conditions, they shunt off things into the secondary growth chemicals. And so probably, you know, we have these debates often. Our breeding programs, we really put them, we've been breeding and selecting, you know, for 100 years at least in these luxuriant conditions for growth. So we've selected all of these traits for yield, which is fantastic. So, you know, why weren't we breeding in really stressful environments? And you can, our evidence suggests from several crops, you can uncouple those things. So if you did that, you can find, because we know from ecology, plants have to adapt to environments. There are winners and losers. So we need to like refigure breeding, and it's a little bit what the organic community are doing, and thinking about climate change. We can uncouple some of these things. What are the targets? I don't know what the targets are, but we do know that we can start to uncouple these things, and we just did it inadvertently for 100 years. That's my take on it.
And do you have any experience of using the sort of indoor environment to manipulate some of these important metabolites, rather than taking a genetic approach? And do you see that's being feasible?
Yeah, because absolutely. So, you know, we were always wanting to make a claim about watercress grown outdoors. It's an amazing food. It's full of glucosinolates. It's anti-cancer. But the problem was that trait had such a low heritability. Like one week it was switched off a bit more. So if you measure the antioxidant status, all the glucosinolates of watercress you're harvesting from a field, it's like all over the place, and you're thinking, we could never make a claim about that. So now we just need to figure out the conditions. So obviously we can breed it and make it much more robust, but if I can find the sweet spot in the blue light or the red light balance or the temperature or the day length or this or that, yeah, I can use any old genetic material, can't I? I'm doing myself out of a job here. There'll be no plant breeding.
Thanks. So we've got a question in the chat from Catherine Freedy. Catherine, do you want to ask that, or will I ask it on your behalf?
I was just interested in running the vertical experiments as opposed to the standard controlled temperature experiments. Have you seen big 3D spatial effects that would impact the design of them?
Do you mean, are some architectures more suited to these vertical systems than others?
Well, so I put my statistician's hat on. Essentially, I'm interested in the design issues around and whether you see strong, so we know within a controlled temperature environment, there are strong trends across, even in a controlled environment. And I was just interested
Well,
to
in
see
my
whether there were different issues.
Yeah, so we have this 40-foot container and it has various panels. So we block the experiment in the way you would do a field experiment. So we have the panels are blocks and we have them replicated. So we have a sort of split plot design. But in my experience, and I've worked in controlled environments and so forth, I've never seen consistency like it in this particular container, like edge effects. Okay, in these panels, you know, you have like 2 edges and 2, like you can see on my slide if that's still showing. But it's so controlled. It's really cool the way the nutrients trickle down, and so I'm seeing a lot of standard response. It's like a joy to work with, actually. It's interesting. But then the genotypes, then when you see these really different architectural 3D things, that's really interesting. So basically, you talked about like positional effects, like top and bottom, or the LEDs are so like uniform, you know, vertical. It's just really fun system to work in. It's so good because it's so uniform. That's my experience.
Thank you.
Okay, do we have any more questions before we wrap things up? Derek?
Just one wee last one. Do you think, my gut feeling is the vertical farming or controlled environment technology advances has really thrown a bone back to the grower. So it's kind of taken and pissed off the geneticists, to be brutally honest, because you can really, we still don't know what the limits of that box are in terms of light, nutrients, temperature, and you can really transform the crop you normally grow into something very different and create essentially a new product. And we're really starting to see whether it's visually different, taste, or actually changing what your nutrients are. I mean, for the vegans, you can add vitamin D into the nutrient system and get an uptake in there and create something very new that will be very expensive initially and become the norm later on. So I think industry's not on the back foot, but I think they're overwhelmed a wee bit.
I would say overwhelmed is a good way to put it, that there's a lot of, you know, you can tell it's a very young science, and it's like changing the paradigm. It's sort of it's like a disruptive technology that we haven't quite figured out how it's going to bed out yet.
Mm-hmm.
So, but going back to like the breeding's out of business, and that's not the way. A number of seed houses now are having a bespoke business for indoors, because some of these companies who are growing indoors now, they're using so much seed, and so the seed companies are reacting to that. So there are 3 companies I know at least who have these special bits for indoor growth. But you're so right. The like, if you call it the indoor agronomist, we accidentally created some like white watercress, and it was terrible. It was a disaster, obviously. But I was thinking, hey, can we market this? It's like that white asparagus that the French go on about. You know, have we got some? And I was trying to persuade my grad students to eat it and taste it, but they weren't keen. But anyway, so you're right. You've suddenly got this thing. It's like this monster where you can create all these really very, very interesting plants that we couldn't in the past. So it is very exciting. Yeah.
I guess some of the seed or breeding houses will be searching through their old varieties for stuff that wouldn't survive outside, because they'll also come with different tastes, textures, performance, and hoping against hope they've not binned it because it will survive in controlled environment.
That was, I was in a debate about that very recently. Think of all the germplasm that companies binned, which was probably extraordinarily suited to these clean environments. That's exactly what the big companies are doing. So Bayer spun out Unfold, Sakata's got a big indoor, and you can imagine that they are just, you know, going through their germplasm and looking at. And also the industry is becoming a bit more, you could say, vertically integrated, in that they're doing their own thing too.
Yeah.
So, you know, because they are the best sort of arbiters, if you like, for screening. So you also see a number of indoor companies owning their own breeding programs because the industry is not delivering to them.
Yeah.
So yeah, it's interesting times.
So I've got a couple more questions come through in the chat. There's one from Robert Rintle. Robert, do you want to ask that yourself, or if I don't hear from you, I'll ask it? Okay, so So the question is, why have you focused on blue light, and have you considered any other colours of light treatment or even UV?
Good question. Well, we're just lemmings, aren't we? So I don't know. When I was in the UK, I knew of some ongoing research on like blue light and shelf life. So that sort of was what started it. And then if you look at the literature, it's blue light that's been manipulated. But we are desperately keen on manipulating other aspects of the spectra. And actually, I was in a company indoor farm just recently, desperately trying to look at the LED lights and recipes for lights are probably the biggest tightly held secret by the commercial entities. So no, it's a big deal. I think there is so much more to be done there. So we're in discussion with this company to make our system a bit more malleable for very specific wavelengths, and that will be super exciting because we know how these, blue light photoreceptors, we know they impact stomatal behavior, and of course they're going to be really important. So absolutely, it's an area for the future. We just started with the simple stuff.
Okay, we've got another question from Catherine. Catherine, do you want to just go ahead and ask that?
Yeah, it was just, it struck me that if you're getting such a difference in form and nutrient and just general character through manipulating conditions, what do you think the implications for IP on commercial wines would be in terms of DUP? Because they're neither uniform nor distinct.
That's a really, really good question, isn't it? Because those trials rely on that, put it out in the field a lot, and you can come up with these distinctive characteristics. Like many things, it goes back to this idea that we're a little bit in the Wild West. I guess distinct, you going forwards, if you sort of had release a variety, there would be a set of conditions for which distinctiveness holds, maybe. Because those distinctive traits were just about introgressing traits and fixing them and in a number of environments, so we can redefine that in a way. That's a really good question. Yeah.
Okay, so I'm going to give people one last opportunity to ask any more questions. No, so thank you again for an excellent talk, Gail. It's been really interesting and stimulated a lot of discussion, and hopefully will keep us thinking for some time in the future.
Bye, guys. It's been a pleasure.
Thanks.